Okay, thank you everyone. Good morning. It's nice to be here today. This is a very nice session. I think there's a lot of complimentary talks here. I started in Mote about 2 years ago, and I received the privilege of taking up a project that had been established long before my arrival. The data go back as far as 2003, and continue mostly uninterrupted until 2009, and then there was a little break there, and we picked it up again in 2013 and collected some more data. So the purpose of this project was to sample, originally to sample 4 different creek types on the eastern shore of Charlotte Harbor. 2 of the creek types, seen in this top plot here, northern creeks, were chosen because they represented more natural systems, that is, less altered systems. With the exception of, they're not evident too well in this picture, but there will be another one. With the exception of old mosquito ditches, this was pretty much original hydrology. In the southern 2 systems here, the southern 2 creeks, these are taken to represent altered creeks because of the changes that you can see very apparently right here, representing altered hydrology. Obviously, had the increase in impervious surfaces at the drainages of these 2 creeks. I want to mention this was originally started by Aaron Adams back in 2003, and he coupled this work with several other types of projects, complementary work, mainly focused on snook in these creeks, but entire fish communities were assessed. So, these are the north creeks, the unaltered, so-called unaltered creeks. They branch off here into the north and south silcox. And the Yucatan and the Culvert are those 2 lower creeks. Now, most of the data that were collected in silcox creek systems up until 2009. These creeks here had data collected in them also. There was a little bit less effort. Funding sources were different between these 2 projects. They were sort of joined together. The silcox system studies last a little bit longer. But nevertheless, it's a very extensive data set. The methods used here were to seine in different sections of the creeks, divided into 3 zones. There were 5 seine samples collected in each one of these zones. The seine is 23-meter seine, 1/8-inch mesh, so it really does a good job at collecting everything that's of interest in these systems, and then some. Mud and sand can be burdensome at times in some parts of these. And so every other month, the seines were dragged in these 3 different zones of each one of these creeks. And all taxa were documented to the lowest practical level. And individuals were measured, the first 25 individuals were measured. So here's some pictures of what I just explained. The seine is deployed some distance, about 10 or 15 meters from the shoreline. Sampling was done at a low tide too, to counter the effect of a fish that may have the opportunity to avoid the seine by getting up into the mangrove roots at a higher tide. I don't think that difference was ever really tested, but it was just based on a logical assumption that would be the case. So we would collect the fish and bring them over to a canoe that would paddle to get in, access these very low water environments, count our fish, ID our fish, and collect the standard multimeter water quality data, temperature, salinity, oxygen levels at each one of these sites. Okay, so I want to really explain a little bit more about the history of this project and the amount of data that were collected and the various reports that were produced over time and the restoration that occurred during this period. Silcox creeks being natural creeks. But nevertheless, there was restoration done over time. These mosquito ditches in the northern part of the creeks were scheduled for restoration, which was conducted in 2008, completed, I think mainly during that same year. There's about 9 miles of mosquito ditches that were backfilled, and that covers some 350 acres, I think. So the point of this was to restore that natural hydrology we all think of, the sheet flow rather than the channelized effects. Okay, this is just a summary of what I just mentioned, the pre-sampling era. It can be seen as the 2003-2007. Some data collected in 2009, and in 2013, just last year, we sampled April, July, September, and November. Okay, this was something you don't really have to pay attention to the details. This was my, for my benefit, when I was going through the vastness of this database and understanding what was done when and what reports were produced over what sections of the project, and get some idea of what the methods and results were. I collected none of these data. Taxa were described functionally by resident, juvenile, or transient. Essentially, what we're looking at is the abundance and diversity, a typical approach to this. And abundance was standardized by density. So number of fish per square meter was how things were standardized and compared. And over this time, from 2 different periods, 2003, 2004, and 2006 and 2007, the main result was that the 2 natural creeks had higher abundances overall of fishes. And the snook, which was a focal species of most of these studies, was too variable to really be used as a valuable indicator. So there were more data collected, again, in 2008, 2009. There was a little bit more analysis done with this because there were nutrient data incorporated into the study and water quality results associated with it. There were lower overall density of fishes from this period, which was explained by very low rainfall. So that aspect ties in with what we have heard already about this rainfall really driving the fluctuations in these populations. It's overarching. So this was probably confounding to understanding how the restoration that occurred may have impacted these populations. But nevertheless, it's fortunate because it's part of the natural variation that's always going on in the background, and it's so difficult to separate from the intended consequences of this work. That is the restoration effort. Something that is interesting about the snook again from this project is that the Culver Creek, which is one of the southern creeks with the most altered drainage patterns, actually had the highest densities of snook for one of these years. So anybody who's been out collecting these snook know that you'll find them in water you wouldn't even want to walk through. They can tolerate some pretty nasty conditions. Okay, so along comes 2013, and I get this opportunity to go out and be in these creeks for myself, and I was amazed because having spent a lot of time in Charlotte Harbor, I never spent time in these specific environments, and that really is a great place for people to experience the value of these habitats and the importance of all this protected area along the shorelines of Charlotte Harbor. This is where you find snook and redfish as big as your fingernail. So that's really the background of the effort that has been put into this study. There's a number of years behind this. There's a large data set. I spent most of my time so far going through this data set and understanding how it was built, how it was established, how fish were counted, and mainly to what level they were distinguished taxonomically. being lumped. So I've got some analyses, just very basic things that show some trends for some of the more abundant species. And so Brandon, there's some Gambusia patterns in here that I think you'll recognize as maybe, I think, being contrary to what you showed. We have Gambusia, the mosquito fish, in blue, and the way these are calculated and expressed is the mean abundance for each season for each year. So the wet season's on the top. This is the North Silcox Creek, one of the natural ones. And the dry season is on the bottom. Trends in abundance for each species are pretty similar between seasons, but as you'd expect the amplitude of the y-axis changes. In the wet season, the rainwater killifish, the parva that's in the red, is really the abundant species throughout the period of this study. The mosquito fish starts pretty strongly in these early years, but it demonstrates a continued decline almost to the point of extinction in these creeks. I think I found 10 of these fish in 2013. It's interesting to note that. I don't know exactly what it means. These fish have a very wide tolerance to environmental conditions. We have fluctuations in flood and drought going on throughout this period, and a number of other factors going on in the background. The South Silcox Creek is demonstrating the same trend over the entire period when compared to the North Silcox Creek. The same fish are the big players, but the overall abundance of these species is lower in this creek. Okay, just to show you the species everybody likes, the snook and the redfish. Generally similar patterns, more abundant earlier in the study and a slight bit of a decline. I realize I forgot to put the error bars on here, so you can't really tell quickly by visualizing who's significantly different from who, but the standard deviation of these catches is really quite high. It's as high on the y-axis as the means are in many cases. So there are a lot of variability in these densities. But the trends are pretty similar between species. This is the South Silcox Creek. There is a large pond at the South Silcox Creek that's connected by a very narrow stretch of shallow water. And that pond is home to a great number of snook, and that's really why these data are demonstrate a higher abundance of snook in this creek. In fact, Aaron had done a number of studies in this particular pond. It was just beautifully set up for looking at growth rates and mortality and putting up an antenna system similar to what people have done in the Pacific Northwest to look at smoke going through the rivers there. Okay, so that's pretty much the background, and I just have some basic data at this point to show. But in the future, the analysis are going to be similar to what Brandon showed with his talk, using primer to look at a lot of these patterns of the entire community, not just the overall abundance, even though the overall abundance is what previous authors have suggested is the most important metric to characterize the value of these creeks. But we need to look at the changes in the communities under different conditions to understand some idea about how restoration efforts impact these communities, and couple that with background of natural variation, that we can tease out differences between as much as possible. Okay, thank you very much.
Oh, okay, good. Excuse me. Jim Cooper, Olympic Conservancy again. I'm familiar with Dr. Adams' study because I listened to one of his presentations. I'm a little bit not familiar with the term altered creeks in the 2008 restoration details on that?
Sure.
It seems like the restoration wasn't all that successful, what you're telling me.
Well, I don't want you to have that opinion necessarily.
Okay.
Because at landscape levels, these things become very complicated very quickly.
Sure.
And inherently variable systems, highly dynamic systems like these, you don't have shortcuts to understanding these. You need long, consistent studies to be able to separate natural variability with the intended consequences of restoration. That's why so many people work so hard for so long to try to understand this. But to answer your question, let's go back to this slide. Look here on this panel over here to the left. Okay, these two creek systems are very similar in their morphology to these creek systems over here. Perhaps these are larger and a little more developed, but their morphology is the same. But once upon a time, somebody decided this was a great place to do something like this. And there's the alteration, okay? This is just a classic little example of changes to impervious surface area and so on, okay? We deal with that on a much broader scale throughout Florida. This is as it is today, as you see at this photo. There's no alteration, and there's no restoration. Okay? Let's face it, if we really want to restore Florida, we all have to leave. Okay, the restoration that I'm referring to in 2008 is relic mosquito ditches built in the 1950s. Okay, there's I think it's about 9 miles of them. Okay, and you see that the restoration is to fill them in, and so just backfilling these in, you end up later with something that would be, as was pointed out to me previously, salt tern habitat, and that's one of the primary goals of that effort. Okay? Now, in terms of hydrology restoration, that is to promote the sheet flow, filter marsh, etc. Okay, so we don't have these rapid pulses of water and the associated nutrients hitting these creeks. Nutrients are a good thing to a point. So that's the restoration that I was referring to.
It doesn't seem like the wetlands increase in size very much.
Are you referring to these barren areas? Or
Well, I'm looking at the little lake systems
up here?
Yeah. And if you fill the ditches in, I'm sorry, if you fill the ditches in, how are you making the sheet flow work better unless you do something to hydrologically connect those things?
To connect the ponds? Or
Right, to make sure the sheet flow is continuous and you get more of a flow, more of a natural slow flow.
Those ponds are not man-made.
Okay.
That's the typical process of carbonic acid forming in limestone that creates dissolution and opens these up. Look at this one over here, okay? That has a little entryway. We sampled a lot in there. I had to get in a canoe and duck down through the mangroves, and sometimes the water is that deep. It's loaded with snook. It's also got a lot of redfish. These snooks seem to just kind of keep going upstream as far as they can until they hit these dead ends and stop. So somebody asked the question before, what would you do if you were the king and you could restore these habitats? Well, I think that these connections, although not necessarily, I'm taking the liberty of asking myself a question is to, I don't see a problem with connecting these big ponds, given what they're used for. And if there are connections, I'm not so sure that filling those particular connections in is an advantage. But, that requires a lot of data over time to tell.
This used to be one of the great waterfowl areas of America, and that's why Ding Darling came here. And at that time, these ponds were not connected. And these were the places where the ducks and the waterfowl liked to be, and they didn't like them being connected with places where large predatory fish and things could come up with. But my point here is that anytime you're looking at snook in this area, in the period of record, you got to remember the great snook kill that happened with the freeze. So snook abundances are going to be heavily affected, even for small snook, following that big killing freeze that we had that really killed most of the snook we had in Charlotte Harbor at that one time period. So as you examine a restoration, you got to account that there are other epidemic factors which were affecting snook abundance separate from your restoration.
Absolutely.
And the function of this restoration was not to make snook in the tributaries, but to make salt tern, which is one of our rarest saltwater habitats we had in Charlotte Harbor. 80% of that was destroyed with the kind of development he showed in this slide on letting up Japan in those places. They built them into salt terns. So this is interesting, and I think it's a very good point. You've got to continue the study much longer term, and maybe look at more measures of success than just one target species that admittedly is charismatic and people make money off of, but may not be the whole point of the estuary.
Thanks for your comments. I think there's a couple things that we need to clear up there with some of those. Though snook are not the focus species of restoration. Snook are one that are just used as an example. The focus of the restoration is the fish community, specifically the prey species, the species that are trophically important at a lower level. The freeze is important for sure, and that's one of the obvious points of natural variability that I was discussing. So I think there's a balance. I mean, the salt tern habitat and the history of the environment is what a lot of people are focused on, but there are also other values, and they're not unrelated, and they're not disconnected. They're all connected. So I don't think we look at this as this habitat is valued more over this habitat. These habitats are holding a place and value together, and they are connected. And that attitude it goes beyond just this small part of it, like Phil was mentioning this morning, the connection all the way offshore. So it's a wider perspective that we have to keep in mind when we're thinking about the effectiveness of restoration and the approach to it, I think.
Can I just add something?
Thank you very much. It's been an interesting session.
In response to Jim's comments, we understand that the snook are not the target species of this restoration project. It's interesting to go back after the fact and design a monitoring program that's supposed to measure what you're trying to measure. In this particular case, we went after the fact to figure out how the habitats responded to the restoration and filling, the hydrologic restoration and filling of the mosquito control ditches. We were looking for measures of changes in function and structure of those wetland systems along the shoreline. So since we didn't plan this ahead of time, we didn't have pre-restoration parameters that we could go back to, except in this particular case, we were fortunate enough to have this long-term data set about the juvenile fishery response. So that gives us an idea of if the system is functioning at one piece of the total component. And so that's going to be matched with some habitat measures if we can use aerial photography to see changes in wetland vegetation and look at some changes in flow and water quality. So Jim has done a really good job in taking the historical data, historical, 2003, taking the past data and using that to capture a baseline for which we can compare our meager amount of post-restoration sampling to. So don't take this out of context. He's done a really excellent job, and we're well aware that it's one piece of the puzzle. And was it Dave that showed the puzzle slide with all the puzzles fitting together? This is one puzzle piece. So we're really excited to take a look at the data in more detail. Thanks.